Yesterday was a pretty good start. Today will be even better. And David promised us that tomorrow would be the best. If the past is any indication of at least the immediate future, David will deliver. We heard yesterday that such a statement is a case of induction. To reason from experience means acquiring information and detecting regularities. So think pigeons as art critics. The anthropologist, I think it was Gregory Bateson, the anthropologist, who said that information is a difference that makes a difference. Information starts, that's what he meant, with being able to make a distinction when there was none. Unfortunately, that is also how hallucinations start. So making a distinction is therefore not enough. It has to be a distinction worth making. But therein lies the rub, because it is not anyone in particular who decides worth in the longer run. So in 1999, I spent a year at the Institute for Advanced Study in Princeton on a leave of absence from the Santa Fe Institute. And that is where I first met David Krakauer. At that moment, I knew what it must have been for explorers to first encounter a platypus. An egg-laying, venomous, duck-billed, beaver-tailed, otter-footed mammal. What the seemingly paradoxical cross between half a dozen species is to naturalists, David is to intellectuals. David is an intellectual platypus, and that is an endangered creature in a world chasing the next tiny idea jointly recognized as fundable by the NIH, the NSF, and your peers. Luckily, a zoo in Santa Fe, known as the Santa Fe Institute, recognized the treasure and captured the specimen. That zoo does not exist without our and your support. So if you consider it a worthy cause and can make a donation, please consider doing so. Wittgenstein thinks of genius as being talent exercised with courage. And the talent of David is obvious in these lectures. But it is David's courage that permits him to attack questions that hinge on integrating disparate, and at times perhaps desperate, domains of knowledge. David's perspective, however, has a precursor, and it goes under the name evolutionary theory of cognition, or more generally, evolutionary epistemology. And one of the major architects of evolutionary epistemology was the late ethologist Konrad Lorenz, who was honored in 1972 with the Nobel Prize for his work on animal behavior with Jan Tinbergen. Somewhat perpendicular to Kant, Lorenz thought that the world is knowable, but it is knowable through the categories of the knower, which were shaped by evolution. So evolutionary adaptation by natural selection results in a partial correspondence, a kind of isomorphism, between the structure of the world and the organization of the knower. On that account, organisms do not make theories of the world. They are theories of the world. David Krakauer rightly reminds us that this perspective needs to be revisited, and the time is ripe to make it more rigorous, quantitative, and formal. Let me quickly close by quoting Bateson again, that information is a difference that makes a difference, because this not only aptly describes our fast-paced time, but it pretty much summarizes 4 billion years of evolution that got us here. Cells acquired ever more sophisticated molecular machinery to refine old and make new distinctions about their world. Cells learn to distinguish between carbon dioxide and glucose, day and night, self and non-self. But for all these molecular sensors to make sense, they must be followed up by an information processing machinery that determines actions. And for these actions to be worth making, They must improve the Darwinian bottom line of the actors, but there is more. By virtue of their actions, observers change the very world they observe, thus giving rise to potentially new differences that might become worth detecting. It is this feedback that turns evolution into both an information gathering and creating process. An information race that gave rise to vast molecular and cellular networks, cellular interaction networks, in fact, the internets before the internet. And David's second lecture will illuminate this process. David.
All right, here's the platypus. Notice that the machines are on my side. The interference was going to swamp his comments. I should say, when Walter was being made a professor at Harvard, one of his letter writers said, to not hire Walter would be like not hiring Wittgenstein. And so you perhaps should get that, not me. Okay, so good. So as Walter said, tonight will be a sort of interlude between an exciting first talk and a truly exciting third. So let's just start with some action. Again, I'll use that, little warning icon when the goings get tough. Right? That's Prokofiev, The Dance with the Knights. And that's a real tool-using octopus. It learned to do that over its lifetime. They use coconuts the way that we would use a shield. And I put that up at the beginning just to remind you of where I stand. And that is that I believe that cognition and intelligence is ubiquitous. It's present across nature and at multiple scales within ourselves. And today we'll be really emphasizing that multi-scale cognition within the organism. But I need to introduce you to how we've thought about the evolution of intelligence in the past. Yesterday I started with some crazy anthropological theories, and I'll now show you the current state of the art in science that tries to account for how it is that we use iPads, right? How it is that we use language as we do. What is it, right, that you could extract from the basic anatomy or genetics that might be a clue to what we think as exceptional human characteristics? And so the obvious thing is that we have such huge brains. It doesn't take a scientist to recognize that Albert Einstein's brain is significantly larger than the brain of the mouse lemur. And whereas Albert Einstein introduced us to curved space-time, the lemur eats grapes in a forest. And here are the two brains drawn to relative correct scale. And so for many years that was considered the critical, if you like, anatomical correlate of our exceptional capability, just larger brains. And it's not totally stupid, because larger brains correlate with a larger number of nerve cells, brain cells. And so if you go from very tiny little brains down there to that huge human brain over there, down here what you have is a number representing the number of neurons. And so it's not just a naive assumption that the larger brain is associated with greater intelligence. There is, in fact, some, if you like, mechanistic justification if you believe that the cells in the brain contribute to the brain's abilities. Oops. If you just look at the size of brains in nature, unfortunately, we don't have the largest, neither do we have the largest number of cells in our brains. And so it can't just be that, because if you're trying to account for traits that you think of as uniquely human. I mean, for all we know, the toothed whale does all sorts of things that we don't appreciate, but we're trying to account for culture, symbolic language, music, mathematics, and so forth. So that doesn't seem to work too well. That might be naive, and so an alternative approach is to consider what's called the encephalization quotient. That's the ratio of the brain to body size. So, for example, you might expect a whale to have a larger brain, because after all, it has a larger body. And so if you look here, his body size, body weight, against brain weight, and there's a trend. This is called a scaling law. You've probably seen too many of these going to Santa Fe Institute talks. But you'll see that there are points that lie on the line, and then there are points that buck the trend. So the bat, for its body size, in fact, has a smaller brain weight than you might expect. So based on this kind of reasoning, you'd think the bat wasn't very intelligent, okay? And as you go up here, if you look at the blue whale, that's rather like the bat. Even though it has a huge brain, it's below that line, and so it's lower than you might expect given the trend. And so you look at humans and you say, this is fantastic. Look at humans. Not only do they have big brains, but they're way above the line. Oops. So's the dusky dolphin. So that doesn't seem to work too well either. So people have looked much more carefully at scaling within the brain, not just the brain to the body, but parts of the brain that we know are associated with what we might call higher cognitive processes, and parts of the brain that are not implicated in those kinds of processes. So, for example, looking at, I'm not sure you can see this at the back, the prefrontal cortex that is associated with strategy, decision-making, and the neocortex, which also is involved in higher functions, but not quite as high, if you like, as the prefrontal cortex. And what we have here is the neocortex area against the absolute prefrontal cortex area, or the relative. And here, when you look at the primates, notice no whales on these plots, the humans do come out rather well. So, in terms of relative brain regions, there does seem to be some evidence that humans are different, at least from the other primates. Now, people now have also, of course, looked at genetics, so genes associated with difference. And so how does this work? What we do is, this is a tree, what we call an evolutionary tree, and we're looking 20 million years back. Now, if you look at the rodents, the rat and the mouse, their common ancestors 20 million years ago, and yet their brains remain more or less identical. This is actually the ratio of the brain size to body size, the ratio of the encephalization quotients, doesn't matter. Whereas if you look here, at the primates, in the same time, 20 million years, there's been a significant divergence of our brain from the macaque brain. And over here, what this is it gives you, if you like, the rate of evolutionary change. If you look at housekeeping genes, genes that are present in all cells, there's no difference between the rodents and the primates. But if you look at genes that are expressed in brain tissue, it looks as if the rate of change has accelerated in the primates compared to the rodents. Okay? But note that this is true for all the primates, not just us in relation to the other primates. This is the most recent result, and in fact, one of the authors on this paper is Michael Lachman, who was a former postdoc at the Santa Fe Institute, who now works in Leipzig. And this is quite complicated, this picture, but what you have here is you have many, many, many genes, and you're looking at their rates of expression. And when it's red, that gene is more highly expressed in a human, and when it's blue, it's more highly expressed in a chimp. And you have different tissues, so this column would be brain tissue, heart tissue, kidney tissue, liver, and testes. And you can probably already see that if you look at the differences across tissues, the only tissue that shows a significant change in expression are the testicles. Okay, so, which is a little bit alarming, because we really, really wanted the highest part to be over there at brain. So what I'm telling you is, based on this evolutionary evidence, at least over a period of about 80 million years, there doesn't seem to be a very significant difference that we can use, if you like, to explain differences in cognitive function. And yet,
what a piece of work is a man. How noble in reason, how infinite in faculties, in form and moving, how express and admirable in action, how like an angel in apprehension, how like a god. The beauty of the world, the paragon of animals, and yet to me, what is this quintessence of dust? Man delights not me.
Right, but you can't help but be delighted by that, by Shakespeare, right? So, I mean, there's something that we have to account for here. And so I want to broaden the evolutionary question, go further back in time, further than the 80 million years, right back to the formation of our galaxy. So, our solar system is about, formed fairly quickly after the Big Bang, so I don't know, 13.5 billion years or so. And the Earth accreted out of the solar nebula about 4.6 billion years ago. And then within 800 million years, a relatively short period of time, life appeared on the Earth. Simple form of life, like bacteria. It took, you can't see it in this slide, about 2 billion years for those cells to form the cells, to evolve into the cells that we're made from. Eukaryotic cells. These are called prokaryotic cells. And then another 800 million years to get jellyfish. But the wonderful thing to realize is that once you had jellyfish, you had Darwin, you had Isaac Newton, you had Murray Gell-Mann, right? The long delay in the evolution on Earth wasn't in the formation of things that we view as uniquely complicated, but in fact, the evolution of a cell. And so what I'm going to be talking about today is the extraordinary power of the cell, a little bit along the lines that Walter mentioned. Because if we understand that, I think we'll understand intelligence. Here's another way of saying it. This is just a spiral representation of history with time unfolding up as we move up through the spiral. There you see the 4.6 billion years of Earth history, and the longest delay that I mentioned in going from these bacterial cells to eukaryotic cells, cells with a nucleus, with an internal membrane and chromosomes that segregate independently. So let's talk about cellular intelligence. And you already know what I mean. I mean representation, I mean inference, I mean strategy, and I mean competition. I mean the possession of those basic ingredients of intelligence. I do not mean high IQ test. Okay? And so here's what you might think of a sensory system mediated by a nervous system. So here's someone eating what I assume it could be a cigar. It's more likely that it's a sausage. They're looking at it and they're smelling it and they're tasting it, and that's being processed by a whole range of specialized cell types. But a cell has a similar structure. On its surface, it has receptors, it has pathways that act a little bit like sensory pathways in the nervous system, and these all get integrated into the nucleus that looks a little bit like our neocortex. So there's a structure to the cell that resembles, at least superficially, the structure of the nervous system. And, in 1965, these two gentlemen, Monod and Jacob, were working on the internal circuitry of E. coli, a bacterium. And this is the origin of this field that you might have heard of called gene regulation. That is the internal circuits that turn one gene on and turn another gene off according to inputs from the environment. Okay? And these circuits lead to quite extraordinarily complex behavior at the level of single cells. And I'm going to show you a video, and what you're going to see is a eukaryotic cell, a white blood cell, chasing a bacterium surrounded by red blood cells. Oops. That's the bacterium there. Okay, triumph of the eukaryote. All right, and so it was caught. Now, if I showed you that, video, you say, well, that's an animal. I can't quite see. It looks a bit pixelated, but it's one animal pursuing another animal in some complicated environment. And because, of course, it had to sense where it was, it had to pursue it, had to finally apprehend it and consume it, and the E. coli was evading it, and so on, right? So it looks like a form of intelligence. It requires strategy, requires representation and inference, but there are no nerve cells here. These are single cells. So each of these cells has in them, along the lines I said, a complicated molecular nervous system at a lower level that has nothing to do with nerve cells. So I'm going to spend most of my time talking about cells in tissues, the capabilities of cells in aggregate, not on their own. And that leads to all sorts of complexities that you will probably start to think about. But what I want to show you is that when you probe the capability of the single cell, you observe extraordinary representational power. And so, again, just for those of you who weren't here yesterday or those who were, I want to make this contrast between what I call the dumb cell. The dumb cell is a bit like an integrated circuit or a transistor. It doesn't really do anything, as opposed to the intelligent cell. These are real cells, by the way. These are nerve cells during the development of your brain trying to find partners. And they're moving around and they're probing axons to determine whether they like them or not. And if they like them, they'll envelop them. Otherwise, they'll leave them alone. And so these are how real cells in your body, in your brain, behave. They don't behave anything like that. In other words, they don't look like our idealized computational model of the brain, which is wrong. So I'm just going to give you an example of what a cell can do. This comes from some of my own work, and I call it cellular perfect pitch. And for those of you who don't know what perfect pitch is, let me show you.
How about this note? Ooh. E, that's right. How about this note? Ooh. Hit it. You are right.
A-flat.
That's an A flat.
Yeah. 3, right? Now you think, wow, that kid, well, one cell can do that, right? And so this is what happens when you estimate or guess a note. Sound is transmitted as an oscillation in air at a certain frequency. Higher notes oscillate faster than lower notes. And when you hear them, your brain processes that periodic signal, and you conclude that it's a certain frequency that we associate with a symbol like C or A, B flat. And cells do exactly the same thing. The difference is, instead of it being a sound wave, it's a wave of concentration of a protein released by the brain. And if it's a low-frequency wave, the cells turn on one gene through a complicated circuit that we've worked out mathematically. And if it's propagated in a wave at high frequency, it turns on another one. And the reason this matters is because these little cells that we have in our brains, men and women, they actually mediate the entire reproductive cycle, which occurs at much lower frequencies. In mammals, in female mammals, typically on the order of a month. So all of that complicated reproductive periodicity is mediated by a little cell in your brain that has perfect pitch. And if it gets it wrong, all of that gets messed up. Now, it would be inappropriate to talk about the power of cells if I didn't mention the two most famous cells in neuroscience. And these would be, if you like, face cells, face-selective cells, and mirror neurons, which I'm sure some of you have heard about. Face cells have an interesting history. In the 1970s, a scientist called Gross was probing neurons in monkey brains. And he found cells that responded exclusively to images of hands. He gave them various things that looked like hands, feet, various pads, and they didn't respond, but they only responded to hands. So he called them hand cells. People thought there's just no way that you have a cell in your brain that responds only to a hand. And that led to a hypothesis called the grandmother neuron, that in fact, if that's true, then you might have a cell in your brain that exclusively responds to the image of your granny. And that was considered funny, and it was a bit of a joke until they discovered that it was true. So I'm going to talk about these two different kinds of cell. Apologies to Velasquez. The Rokeby Venus has been brained. And I did this for a reason, because the cells I'll talk about, mirror neurons, not only do they recognize expressions, but they are actually the roots to empathy, to self-knowledge. So this is what it means to be face selective. And so this is recording, I'm not sure how well you can see at the back, this is recording from a single cell, and these are, if you can't see the monkey faces, where slight modifications have been made. So here, a band has been removed, here you've put a human face in, here you've replaced it with a hand, here you've rotated the head. And what you observe is that and what this little histogram represents is the strength of response of firing of a nerve cell. And what you see here is it responds most strongly to this image, and to the partially occluded images, or the rotated profiles, it responds less strongly. And here's a brush. That would be called a control. And up here is a much stronger response, a profile-selective cell that only responds to cells in profile. So this is quite remarkable, right? Of course, these cells exist in the context of many other cells that are giving them information, but at some level, this cell is representing an object, a single cell, as complex as a face. Now, what happens when you remove those cells in humans, for example, when humans suffer a stroke?
In this test, Terry takes a Polaroid picture of her mother.
Terry, you just took a picture of your mother. And I want you to take a look at these and see if any of them are the picture you took or not. They may...
Now, Massachusetts General Hospital researcher Dr. Nancy Etkoff shows Terry several Polaroids, with only faces showing, and asks Terry to pick out her mother.
Okay, if you had to guess, which looks to you the most like your mother? Just looking now, you can't tell which one of these. None of these really look like your mother to you.
No.
Okay.
If you just cover up the face and I just look at the clothes, I can tell you.
Okay, do you want me to uncover them? Okay, why don't I do that? I'll uncover them so you can see what they're wearing and other parts of the picture.
And see, that's her, because the pink and purple shirt.
The shirt. Do you recognize the shirt?
So I don't know if you heard that, it's a bit muffled, but she was unable to identify her mother from her mother's face, but when they revealed the clothing that she was wearing, in a characteristic pink jersey, then she was able to identify her. So she wasn't in any way visually impaired. She could explain to you what the components of a scene were, but she just couldn't get the face, okay? And that's the inferential power of a single population of cells. Now, okay, that goes wrong. Oops. Back.
In this test...
So let me show you another video. So these are two neonates, newly born primates, one hominid and one monkey. And they've never seen themselves, obviously, and they're being exposed to faces. This one across species. And notice what they're doing. They're actually imitating what they're seeing. Now, let me ask you, which is cuter? That one, right? All right, they win out on the cuteness dimension too. All right, no, I'm sure to the parents, the baby is totally adorable. Now, what's remarkable about these kinds of capabilities is that they also are computed by single cells called mirror neurons. Okay? And mirror neurons have been discovered very recently. I'm sure there's a Nobel Prize about to be given out for them. And what mirror neurons do is something very interesting. Mirror neurons light up when they see you do something, like move your hand. Right? But they also light up, let me just get that right. If I move my hand, Walter's mirror neuron would light up. But if he moved his hand, the same neuron would light up. That's what defines it as a mirror neuron, in the sense that it both responds to an image in someone else, when someone else moves, and also it responds to your own motion in the same domain. So they're both sensory and motor. Does that make sense to you? And this is considered the root of empathy. So when you cry because JR died in an episode of Dallas, you think, right? Or you cry because you see someone's eye watering. You think, oh my God, am I really that sentimental? No, you just have a very active mirror neuron, okay? Because your mirror neuron is generating the response that's identical to the image in your brain. single cells. And so here's the kinds of experiments people do. They'll show you images like this. They have an actress, and she's asked to pull faces, and you are asked, as the experimental subjects, to classify them in a multiple choice test according to their magnitude. So here you'd be shown a movie with her pulling that face. Well, is that happy? I don't think so. Is it fear? Yes, yes. Not sad, not, and so forth. And people are quite consistent in how they label expressions. And here's another one. What's that? I don't know. And so on. Happy, that's obvious. Disgust, it's an interesting, complicated one. People still get it quite right. There's quite a lot of variance in response, but they don't typically confuse them. And the interesting point is that each of these images is, in fact, encoded, represented in a different population of mirror neurons. And so what you do is you put people in brain scanners and you ask them to do one of two things: to look at the movie, and you measure their brain, or to copy the facial expression. So that's viewing in white, because I'm sure you can't read it at the back, versus imitating in black. Right? So these cells, if you imitate, they fire more actively. But the same cells will fire just by looking. Okay? But you'll see what's quite interesting here is the magnitude of response is proportional to the intensity of the emotion. So here you get a very strong fear response, right? So if you look at something frightening or you make a frightening face, those cells really fire. But different mirror neurons have different profiles, right? So this is in the temporal cortex, this is in the somatosensory cortex, and here things are quite different. These mirror neurons respond most actively to neutral, right? And this is the amygdala, which is very strongly emotionally implicated, and that actually responds quite strong to neutral and to happy. So you have different populations in your brain which are encoding responses to emotional or affective states in people you look at, empathically. So single cells. So I just want to make this point clear. These cells exist in a complicated circuit of cells, okay? But it could be the case that the way the brain works is that no single cell ever represents the whole. It could be, if you look at the Mona Lisa that's been pixelated for an image on Times Square, that every cell just encodes one of these colored rectangles. It never, ever does the integration where a single cell represents the whole, which they do. Computers don't have this characteristic. If you look at a single transistor or integrated circuit, it only ever represents a part of the whole. Your brain has a very intriguing characteristic, that there are cells that represent the whole, the entirety. Now, there are limits to cells, and so why did we ever shift from being single-celled prokaryotes to being eukaryotes? What are the driving forces in getting cells to aggregate? Now, to answer this question, we look at organisms that have both features in their single life cycle. This is known as a slime mold, and most of the time it lives as a single-celled creature. But when the environment becomes bad, when it dries up, they aggregate and form a multicellular organism. And that multicellular organism can migrate, and when it's migrated to a new area where there's food available, it stops. Some cells form the foot, and they're destined to die, and some form the fruiting body, which will regenerate the next population of slime molds. So there's a clue here that in some sense the reason why you move into this multicellular configuration is to explore a space of solutions that you couldn't as a single cell. And that gets back to yesterday, what I was talking about in terms of intelligence as being fundamentally about search. So here's a video from John Bonner, who's an old colleague of mine from Princeton, who's one of the pioneers in this area, and it's just going to show you that life cycle. They're extraordinary. The way it works, in fact, is this center here is sending out pulses of a chemical called cyclic AMP, and they respond to it by tracking towards it. And that's the aggregation signal. And once they've aggregated, they move into this phase, which you're going to see now. And so this thing is made of many, many cells. Only these will get to reproduce, and all of these will die. It's in fact, you took that footage, I think in the 50s, it's still the best footage of the slime mold sporulation for those of us who are aficionados of this cinematic art. Okay. This is another slime mold. This is its life cycle, slightly different, but this one's kind of smart. And so this one, all the cells come together. They form one giant cell called a polynucleated cell, and then it sends out channels looking for food. And when it finds food, those channels get stabilized, and the channels that don't find food die. Okay? And so you're going to see that now. So these are actually little maize grains. Here's a mold, and you'll see what I just described. So, what one really enterprising researcher did is he took these maize grains and he distributed them according to the major train stations in Japan, in Tokyo. And he said, how would it design the railway system? Right? And what you see actually, this is a solution to quite a hard optimization problem, finding the shortest path between all of these stops. And it came up with a solution. This is the mold network, not to scale, but it came up with a solution that's in fact as good in terms of the distance traveled as that was designed by the engineers, the architects of the rail system. So this is kind of remarkable. It's a stupid little mold. We should use them in little silicon test tubes to do experiments, to do architecture, to design our highways. They'll do a hell of a lot better than our government. So this is a clue to why you go multicell. It gives you this greater inferential power, because what you can do is if you head off in a direction that's not profitable, you'll die off, right? But if you go in a direction that works, you'll stabilize it. So there's that sort of Darwinian flavor again, this exploratory flavor where you do inference by having things die back. Now, I put this in for you. This is a little art interlude, and what you'll see is a maze with a slime mold, and then you'll see a human imitating it. I love it. I was transfixed by that. Slime mold art. All right. Okay. So, the slime mold must be, what an idiot. Not at all convincing as a slime mold. So, all right. So what have I told you? I've tried to give you a sense of how impressive individual cells are, their representational power. And I mentioned the slime molds really to make the point that your brain is really that. Your brain is really lots of those slime molds that are behaving not that differently, right? Aggregated and under slightly different rules of control. And that's what's generating what we consider intelligence. And I'm going to show you one of the remarkable capabilities of our species, and others to a lesser degree, when you aggregate cells, and that's our ability to count. Now, in every conversation I've ever had about human intelligence, people will say, well, it's obvious what makes us special, it's language, because no other species can talk, right? And I have problems with that. Not just with the problem with the question of whether other species can communicate or not symbolically, but the fact it seems to me having a number sense is more important. So if you are, an organism that goes to war, that battles with other troops, you're a monkey, and you don't count correctly the number of opponents that you're facing, you will die. There's no need for communication. You just need to back out if you realize that you're outnumbered. So there is a symbolic internal cognitive manipulation which seems to me more important than the symbolic communication required by language. I'm not deprecating language, I'm just saying that I don't think mathematics sits on language, I think language might sit on mathematics. So let me show you how it works, and this is one of those moments where you get your warning. So out with the text messaging or attention. Okay. Here are two monkeys, Rosencrantz and Macduff. Their real names, and I think I know why they were called that. I wasn't sure. And what you did is the following. If you remember, yesterday I showed you that video with the chimps identifying numbers and getting them in the right order. And so you give them objects in various different colors and different geometries. What they have to do on a video screen is identify either the larger set or the smaller set, right? So you might press the larger one first and then the smaller one, right? So that's ordinality. Just numbers is called numerosity. And what you do is you train them to be able to get the relative magnitude of those sets correct over and over and over again. And so here's what happens. You start them training. This is 1 to 5. You ask how many times did they get it right. And you can see that Rosencrantz starts off ahead, and I think this is why he might be called Macduff. When it comes to the end, Macduff is right there up with him. If you remember, Macduff did rather well at the end of one of Shakespeare's plays. And so maybe that's why they called him Macduff. And what you then do is you test them out of sample. You test them with numbers that they've never seen before, and they are consistently way above chance in their accuracy. These are not chimps, right? These are monkeys. So that's quite remarkable. And how is it done? Okay, to understand that, I need to show you another experiment, and this is how it works. A monkey is asked to look at a point on a screen. You then give it a set of points, okay? 500 milliseconds. You then remove the points and show it the point on the screen again. And then you show it 2 sets, and you ask which one has the same number of points as the original. Now, you get to look at it for 500 milliseconds. It's not easy, even for us. As you'll see, we don't do any better. Okay, does everyone understand? And this is what happens. And so this is a plot, and it says, this is the number of points that you saw at first, and this is your accuracy. So look over here, you see this very broad shouldered curve? What they saw was 24, but they couldn't recall perfectly accurately whether it was 24. They'll often say 16, or they'll say 34. But when they see 2, they always get it more or less exactly right. Okay? And this is just the logarithm of that plot for those who care. It's a very well-known result called Weber's law, exponential scaling in biophysics. But look at humans. Identical. Okay? So again, we have, it's really identical. It's actually a remarkable result. We do really well when we get those initial points projected to us where there are few of them, and we do very badly when there are many. And we do no better than a monkey. So what's going on here? What I'm telling you is, from the brain's point of view, you're no better at representing numbers than a monkey. I said money, which is some kind of Freudian slip when it came to numbers. All right. And how is it done? So what you're looking at here is you're looking at individual cells, and this is the number of objects, and this is their firing rate, how often they turn on. What you see is that here's a cell that responds to 18. Here's a cell that responds to 24. Here's a cell that responds to 30 maximally. Okay? This is called a labeled line code. What happens is, in our brains, in the intraparietal sulcus, and in monkey brains, we have cells that represent number. Okay? This is what this is. So when you see a set of objects, a cell will light up in your brain that says 16. Sort of surprising. So I think this raises a significant puzzle. If monkey brains can do this as well as us, right, or we do as badly as them, how is it that we're able to represent numbers like a googolplex? Right? How can we multiply large primes? What's going on? And I think this is the key to what makes human beings special. So here's the puzzle, okay? I'm telling you that when you see numbers, and this is this Babylonian mind again, it gets projected back into the brain of a human or a monkey, and it gets encoded in the brain as a certain value, right? And if that's true, why aren't monkeys as good as we are at doing mathematics? Because it all gets turned into the same brain code, right? So why do we do it better? And here's why I think we do it better. So here's some number that we see, a number of people. It gets projected back, finally reaching this intraparietal area, sulcus, in the brain. What then happens in us is it gets abstracted and it gets sent forward to the prefrontal cortex. And there are experiments, but I thought they were a little bit technical, so I didn't want to show you, which show how that happens. That gets turned into a motor schema, gets shunted to the motor cortex, which then writes it out in the form of, say, a glyph. That glyph then goes back into the eyes, and that gets projected to a new part of the brain called the visual area. So something very interesting has happened here. Some number has been coded in these very simple cells that we have and monkeys have, but then we do something rather interesting. We actually externalize it in a symbol system and reproject it to a new part of the brain that's different from the part of the brain that the number was first projected to. Okay? Now the question is, why do we have to do this? Why can't we just do that? Okay? This is the great mystery. What I'm going to tell you is, what makes us clever is our ability to shunt representations from one cell population to another so as to make a problem effortless. So this is the human feature. Without culture, we'd be nothing, right? If I just put you in the wild, you wouldn't outcompete a monkey, right? The reason why we all seem so smart is because tons and tons of people over the history of our species have been shunting things into our environment. This is the repository of knowledge that we draw on, not this. And unfortunately, other species don't seem to have that capability to externalize, outsource representation. So this is what we do, okay? This is very special, right? And this is what makes us smart. Now, there's another feature of outsourcing representations which needs to be noted, and that is it's collaborative, and I alluded to that just now. The fact is that if I have an idea in my head, you don't have access to it. If I make a mistake, you can't correct it. But if I go to a blackboard, one of my colleagues is going to say to me, that's wrong. You made a mistake. They can error correct. Okay? That's not something you can do in your head. So this externalization of representations allows for collective cognition. So, no, but not always. So I'm going to show you an instance where it can go horribly wrong.
Look here. I'll show you. Let me rub this out here. And now, 25 divided by 5 is 5. You see, 5 won't go into 2, will it? No. But 5 goes into 25 5 times, you see?
No, you're wrong, Billy. Now, I'm a pretty good mathematician. Now, 5 into 25, 5 won't go into 2, will it?
No.
But 5 goes into 5 once. Now, we didn't use the 2 before, so we bring it down here. Now, 5 into 20 goes 4 times. There you are. 5 into 25, 14.
No.
Right. Well, okay. That's the virtue of externalizing your cognition. I meet many people who make remarks that are no more sensible than that, but you can't really gauge whether or not they're making a mistake until you ask them to stand up at the board. That's why scientists are so ruthless in asking people to expose their thinking processes publicly, as I'm doing now. Now, you could say, where did this come from? Where are the seeds of this capability? And it's interesting, I was talking to Mary Charlotte on her radio show. She was quite right. She pointed out that there is an analogy in nature, and that's what we call niche construction, that there are other animals that do externalize internal cognitive states and collaboratively operate on them. Okay? Now, this is a new area. In fact, a number of us at the Institute, Jessica Flack here, Doug Irwin, and I, have worked a lot on this theory, but we're nowhere near understanding how the creation of these kinds of animal architectures leads into the creation of external symbol systems, and that's an interesting open territory to explore. The point is that for humans, we've created infinite working memory, right? We've created the capability through culture, through external artifacts, of, in fact, creating a repository, a resource that's absolutely endless. Right? And that's, I think, the key to our true intelligence. This is a photograph of Trinity College Library. So as I said the other day, that's easy for a human and a monkey. We now know that. Monkeys can go to 30, so can we. We can't do much better. But that's easy for a human and it's hard for a monkey because it can't do that. It can't write down Arabic numerals, base 10. Okay? So it's not greater numerical competence. We don't have greater numerical competence. We have greater representational competence, right? And we have greater representational competence in the external environment, okay? So it's anchored, and moreover, anchored in geometry. And I'm going to give you an example of how this works now. Oh yes, Stan Ulam intuited this. Before these lectures, I read his autobiography, and this is what he said. It is possible that human thought codes, human thought codes encodes, we'd say, I guess, things not in terms of words, for most people think pictorially, not verbally. There is a way of writing abstract thought in a kind of shorthand. One may call this a visual algorithm. See, I think that's really what we're doing. We might be doing language too, but I think we're doing this first. Okay? And we in fact do this when we build tools. When animals build tools, they're encoding solutions to problems in some sense in physical algorithms. Okay? So here's a beautiful example of this. So I'm going to just recap quickly because we're getting to the end of this tonight's talk. What I'm telling you is what the brain does is it shifts from an area where it has to think hard about a problem to an area where a bit of dedicated brain tissue can solve the problem without thought. Okay? So think about learning a sport. When you first learn how to wield a tennis racket, you have to think really hard about it, and you keep messing up. And in time, it seems to become automatic. And we have a colloquial term we use, we call it muscle memory. There's no memory in your muscles, right? It's neural, it's encoded in your brain, but it's being shunted to a different part of your cortex, which is much more efficient at representing trajectories in space. And what I'm going to tell you is smart people are actually less smart than you think, because what they're doing is they're taking a problem that you have to think hard about, and they're re-representing it so that for them it becomes trivial. So in fact, they're more stupid, right? The key to intelligence is efficiently wielding neural tissue, right? So when you use a computer's graphics chip to do very fast calculations, you realize there's a way of writing code to more efficiently solve problems instead of having to use a regular processor. Intelligence is fundamentally about stupidity. Right? It's kind of peculiar. So here's an example of that. So here's an abacus. Everyone knows what an abacus is. These are called the God beads, if you like. These are called the earth beads. These are worth 5 units. These are worth single units. And so in the up position here, you get a 1, and this remains in the neutral position. And 4, you'd move 4 up. 7, you move that one down to give you 5. 2 up to give you 7. So I don't know if you've all used an abacus. And you can represent numbers and add in very simple ways by just manipulating the beads, the upper and lower beads. And there are master abacus users. Okay? And here's an experiment. It's a little bit like that monkey experiment I mentioned before. I show 2 groups of people a large number. I show them to master abacus users, right? And I show them to people who are competent arithmetically. Okay? And then I put a delay in, and then I ask you, is that the same number as you saw before? Okay? And that's it. That's the experiment. It's recall of a large number. And you take these people and you put them in a brain scanner, and you see something very intriguing. I think for everyone in this room who hasn't been trained intensively in the art of the abacus, the way you represent large numbers is just by remembering the symbols. You say 4, 7, 3, 1, 4, 7, 3, 1. Okay, that's not how an abacus master remembers it. So what happens to us is, so look, we have different brain regions here. green for the abacus experts, red for the rest of us. Okay? So here's a big red area that lights up, okay? And that's the area called Broca's area associated with speech. So we represent large sequences of numbers as symbols that we intone in our own minds. If you look at a master abacus user, they use the visual cortex. They're not actually representing an abacus in terms of sounds in their head. They're visualizing the abacus itself, right? So let me show you a beautiful video which will make this very clear.
Nevertheless, 8 years of hard slog can produce jaw-dropping results. 12-year-old Kota Kizuka is top of the class.
I practice 2 hours on weekdays and 10 hours on the weekend. I want to become the national champion.
Along with the other high flyers, he can now do huge calculations with a purely imaginary abacus, manipulating nothing but thin air. Children like this show us that with enough practice, super-fast mental calculation is possible.
Hi! Hi! Come here, mate.
If you watch the documentary, the instructor says, I only occasionally hit them. But again, just so you understand what's going on here, right, is when these kids are interviewed and asked, how do you do that? They say, I have no idea. It's totally unconscious. I'm manipulating an abacus. I'm not adding numbers or multiplying numbers or dividing. Okay? So what's happened here is, again, this notion that intelligence, fundamentally, at this level, consists in a shift of representation and a shunting to a part of the brain where the problem becomes effortless, because you're manipulating a large part of the cortex which is extraordinarily well adapted to visual scenes. Right? Our visual system is very powerful. If you can recode hard information in terms of inputs that the visual system can comprehend, you could solve almost any problem. Okay? And so when people say they can think geometrically about problems, that's what they're doing. You're doing it the slow, symbolic way, which we're not actually very good at. And they're doing it the geometric way, which we're incredibly good at. So this is the closest I'll come to a definition of intelligence in these lectures. Okay? And it's this discovering representations over which specialized circuitry can infer strategic solutions easily. Okay? Intelligence means not doing things the hard way. So we're sort of at the end, so this is a quick one. So what have I said? So I've said, so humans don't seem to have fundamentally different brains from other species. So that first part of the talk, when you look at all these scaling results, there's nothing that pops out at you which says, aha, the magical ingredient that can explain why we are so different. There isn't such a thing. What we might have is just more cells, okay? The cells, as I showed you with the mirror neurons and the face recognition neurons and the cell with perfect pitch, have extraordinary representational power. So, and you have millions and millions of them in your head. Okay. So multiple cells expand this representational, strategic, inferential capability, right, by allowing for pruning. And so if you just had one, it would be a dedicated representational cell. If you have millions and millions, you can actually dispense with many at very little cost. And when you put all of these together, right, they give us this capability which is really not at all explored. In fact, the two thinkers, if you're interested in this, who I think have thought about it hardest, are really philosophers, Andy Clark and Ed Hutchins. Andy Clark wrote a very nice book called Supersize Your Mind, which is all about externalizing cognition. It's very interesting. But that's something that we do. And what I don't know how to do is go from this multiple representational cell to this capability. There is no theory. I don't claim to have one, and I can tell you no one else does either. So, tomorrow, what I'm going to show you is the ultimate example of the outsourcing of our intelligence, right? Some might claim the complete outsourcing of our intelligence. into the extraordinary representational inferential power of the computer. Okay? And so the talk will be asking these questions. How much of our intelligence gets externalized in silicon? Right? Will it lead to the eclipse of human reason? Right? How will we know, how will we be able to test machines? And there's been a long history, interestingly, in trying to build IQ tests for computers, and they're quite amusing, as you will see. And what are the implications for the future of humanity? I think this is an important ethical question that we should be asking ourselves. When it gets to the point where something else in the world might be in some sense smarter than us, if we use ourselves as an ethical model, it's alarming. And thank you. Good night. Hope to see you tomorrow. There's a question right here. Yes, a huge amount. Yeah, so let me explain the question and translate it slightly. One of the things that people would say about cells is that they have a fixed behavior. You can't modify them, right? Whereas the brain can learn, and that's sometimes called plasticity. Let me just give the example of a neuron. Your neurons in your brain adapt constantly to changing inputs. And so a neuron has been... I don't know how much you know about neurons, maybe you know a lot, but the neurons integrate incoming activity and send out signals in response. And neurons can maximize the information flow from the input to the output, which can be measured using a statistic called mutual information. And that changes during the lifetime of the cell and according to the regularities in the data you feed it. So single cells have huge plasticity, okay? And so the answer is yes.
Can their function then also change? In other words, some of these specialized cells are destroyed and other cells take their...
Well, in fact, I talked about this yesterday briefly. So if you look at the congenitally blind, who are not using their visual cortex for processing visual images, they shunt word representations. And so cells that you might think were dedicated for vision are quite happy to do speech. Yes, at the back there. Can't remember.
It seems in your definition of intelligence that we're talking about mostly unconscious mechanisms. So essentially, at least what I've always thought of as intelligent behavior, high-level cognition, is something that we represent with awareness.
But how do you measure that in other species?
Yeah, no, I know. I have no idea. But my point being that, as you say, intelligence is mostly a point of dumbing down in a way.
See, the problem, I understand what you're saying, but here's the problem. We've had centuries of anthropomorphic theories that put consciousness at the center of the discussion where we can't even measure it in another human. Right? In other words, we know ourselves internally that we are, but we can't be sure someone else is, and as you know, endless ink has been spilt. Right? What I'm trying to do is say that doesn't look at the moment like a very productive trajectory. Okay? I'll let other people do it. And what I want to answer is some slightly more modest questions that don't necessarily bear directly on awareness, where people have neglected the capabilities either of their constituents or of other species. It's not that I don't think it's important, but there are so many people who have thought about it, and by defining it in those terms, you're essentially ensuring that you can never answer it. I mean, that's my point. You can't even answer it in other humans, let alone other species. It's important, but too many people are working on it for me to work on it. Yes? I'm sorry, could I do... Right. So, yeah. So So there are many arguments about what it is, and Douglas Hofstadter, who wrote a wonderful book called Gödel, Escher, Bach, and recently a book called I Am a Strange Loop, is very interested in this notion of recursion, systems that look at themselves, looking at themselves, looking at themselves, right? And that, which we can sort of do, what am I thinking, I'm thinking, I'm thinking, I'm thinking, that kind of thing, right? We can say it in language, and we think we can do it cognitively. And that's the essence of consciousness. There's, I don't know, I mean, at the level of a metaphor, it seems intriguing, but I'm not sure in what sense it presents itself as a logical answer. Again, not much progress on that. In fact, we've been recently working on recursion in non-humans, because it's a very interesting property. But if someone has a way of making that a persuasive solution, really answering the awareness problem, I'd like to hear it. Yes? Yes.
If something comes into the cell, it identifies that piece of genetic material, takes it to another part of the cell. If that part of the cell can't identify it, it takes it to somewhere else.
Yes.
It seems like intelligent.
Yes. So there is if people are interested in this, the person who's done, I think, the best work in this area is Dennis Bray. And Dennis Bray wrote a book called Wetware. And what Bray argues is that it's not just cells that are smart, it's proteins in cells. So he's even more radical than I am, right? And so, and the reason he argues that is related to the question that this gentleman asked, is that there's even plasticity within the cell. That it seems, and this is something that Walter's very interested in, it seems that there are proteins can adopt multiple different configurations, perform different functions. It really does look as if it goes very far down. And the question is, where does it stop? No. Yes, why don't I get this one here? And then Mm-hmm. Mm-hmm. You're absolutely right. That's the question. You're correct. I think they're very related. And so, for those who didn't, So the comment, more than a question, was that the abacus master's ability to manipulate equations or numbers spatially with their fingers resembled the ability of the kid yesterday to manipulate the Rubik's Cube blindfolded. And I think that's absolutely right. And they both illustrate this idea that we're doing computations in a geometric motor domain, not in the domain of the linguistic competence. Okay? And that's why they do it so well. If they had to use language, which is the thing everyone says we do so well, they couldn't do it at all. Right? Now, how is it done that the representation is shifted? It always seems to have to be mediated by an external object, whether it's the Rubik's Cube or the abacus. We don't seem to be able to do it internally, and I don't know the answer to that. Yeah, absolutely, a musician. Same thing again, or an athlete. I would agree. There's right at the back. So is that you, Sam? Sorry. So in both cases they were adolescent boys? Yeah, it's interesting. Yeah. Well, there's two possible answers to that, okay? And so one is relating to this issue of plasticity. So, okay, the question was, right, the observation that the individuals able to perform these tasks incredibly well were children. There were women too. He was interviewed. Well, but I think that's statistically not significant. That was what we call an N of 1. If you looked in that class, it was coincidence. There were girls in that class too. But here's maybe the thing that's not coincidental. So the serious answer to that is, one is that it's true that children have greater plasticity than adults. So that's true for language learning, right? It's true in sport. It doesn't mean that adults are sort of hardwired by any means, right? And there's a confounding factor which has to do with obsessiveness, right? With single-minded pursuit of excellence in a domain of absolutely no consequence, right? Right? And I think that might be the male characteristic, rather than, right? Because if you think about deductive frameworks where we excel, they tend to have that autistic quality to them. It does. It strongly relates to Bobby Fischer. Absolutely it does. Yeah, I agree. There's another one. I'm sorry, the light's in my eye. Yeah. Oh. Mm-hmm. No. So I am a mechanist, and so if you like, I could have given my entire talk in 10 years using chemical language. The advantage of intentional or mentalistic terms is that they summarize concisely all of those chemical processes going on that I could have substituted, I could effortlessly have substituted and had exactly the same meaning. So I think this is a completely misleading belief. And so anthropomorphism is in fact useful because it's a means of efficiently compressing language so that you do not have to give microscopic descriptions. So I believe, if you were to ask me, when we pursue something, we're doing exactly the same thing. We have a whole bunch of chemical reactions that are simply pursuing a chemical free energy goal. It's just that there's a language that has a semantic valence and is efficient that I can use to communicate to you, but I would not make a distinction at all. Yes, please.
What is the role of good memory in intelligence?
So, is that you? Is that Mary Charlotte? Yes. And so, I can't see, you're just a profile from where I am. So this is very interesting. And so, take chess as an example, right? One of the things that very good chess players do is that they memorize, and I'll talk about this tomorrow, huge numbers of plays. There's something called playing in and out of book if you play chess. And so when you start playing chess, every move is in some sense already written down in the Great Book of Chess, right? As you hit the end game, every move is written down in the Great Book of Chess. And so there's an opening game and there's an end game, and they've been getting bigger and bigger and bigger and bigger, right? And it's only in the mid game, which has huge combinatorial possibility, that you're playing out of book. You're not using memory, you're using inference, okay? And why computers have been so good is that they've been, in some sense, shrinking that out-of-game play. Now, good mathematicians, right, have incredible memory for tricks to solve problems easily, right? I'm not saying that's all there is to it, but so I think memory is a critical ingredient and a massively undervalued ingredient in intelligence. In fact, most really smart people I know have very good memories. They pretend they don't, because they think that having a good memory is cheating. But in fact, having a good memory is being smart, I claim. It is, yes, but we're fortunate now that we can all be smart by externalizing it all on our iPads. Right? I mean, one of the advantages of having it internal is we have very, very rapid access. So it looks effortless. But I think it's very important. Yeah. Yes, please. Oh, dear. How would you define it? I know, you a plant. No, I think that, well, so here's the cop-out. It could be, calculating without conscious access. Now, what does that mean? Okay, I mean, there are clearly many things we do without being conscious of them. And so I'm not sure that's really an answer at all. I don't have a good answer. I think it's a very deep question, and I don't know the answer to it. Yes, please.
David, what do you think is the difference, if there is any, between instinct and intelligence?
No. The problem with this is that these are all words, right? So I think that, so I don't think there's a big difference. In the old days, people would say animals have instinct, humans have intelligence. And so in some sense, it was a lesser form. And I think the connotation is hardwired, that not easily modified, non-flexible thing. So I think that's what it connotes, and I don't really believe in it. And so we are, if you like, born with certain biases in how we respond to inputs. Okay? In statistics, these would be called priors, right? And I think instinct, if it has any meaning, is something like the biases we have before we learn alternative ways of reasoning. I don't like it just as an arbitrary demarcation between human and non-human intelligence. Let me get this first, and then, yes, please.
It seems that a large part of our ability to externalize and thus rise above the apes is centered on the visual cortex. So the question is, in those distorted brain body part maps that you used last night,
Yeah.
why were our eyes so small and not much different than monkeys?
Well, I mean, it's worth making a distinction between the processing of visual scenes and the optics. Okay? So
That was a representation of the space in the brain given to...
No, remember though, I wasn't showing you visual, so I was showing you the sensitivity of the eye to touch. So you can make a homunculus for a sensory system. So remember I told you, so if I were to touch your finger, how sensitive, how much neural tissue, do you see what I'm saying? It needs to be mapped to that kind of sensation. And what you were seeing there was the sensitivity of the eye, not the total amount of cortex dedicated to vision. Do you see? Right, yeah, it's a good question. I thought someone, yes, you did, yes, sorry. Oh, got you. The kidnapping of concepts. Yeah. This is the worst audience in the whole world. No, these are sort of the questions, right? So let me expand a little bit on it. So the first honest remark would be that we don't have a theory of abduction. The closest that we come to is a theory of generalization, and I talked about this recently. And that is that you spot correspondences in the structure of problems, and you realize that the inferential processes that you use on A can be generalized to B. And that's can often look like abduction. So you could say to someone, how did you possibly solve that? It looked so totally new and different. Well, it wasn't, because they saw the underlying structural equivalence. Okay? So that would be one answer. Now, in terms of improvisation in music, I used to play jazz as a kid. I know what it means. I really don't know. I mean, there are musicians in the audience. I know there's a composer in the audience who probably would give you a better answer than I would. Would anyone be willing to offer? Oh, okay, so it was a setup. Yeah. Yeah. Right. Well, maybe that. Well, that's interesting because perhaps that gets to this idea that I mentioned yesterday of composition of representations, right? And so, which sounds very much like what you're saying, that there are ways of representing problems that allow you to mesh them together, and recognizing how to translate them into terms that allows you to do it is the key. That's interesting. Let me just take, I know that, did Ruben, let me get one more, let me get two more. Ruben, you're not allowed to say anything difficult or harsh.
You mentioned the importance of stupidity already,
Mm-hmm.
and I appreciate that. But I think there's more to it, because scientists are mainly interested in intelligence, but psychiatrists, therapists, school teachers deal with stupidity a lot.
Mm-hmm.
And it would be interesting if brain scientists and theorists could figure out why there's so much stupidity. I mean, natural selection would lead to intelligence.
Well, it doesn't.
And then we would survive and not destroy the world. But stupidity seems to be stronger. Why is that?
Oh. I don't know. I don't think, you see, I don't agree with you. And I don't think the problem is stupidity. It might be ethics. So you could have extremely, just think about scientists who commit war crimes. This is not the absence of intelligence. It's the absence of judgment. Now, you might say, okay, fair enough, I'm being inconsistent. It's the absence of one form of intelligence, right? And so I want to make that clear, that I don't think that ethics is synonymous with deductive power. Okay. Now, the other point, though, stupidity in society and what makes individuals stupid, I don't think it has anything to do with Darwinism or natural selection. I think that we're all, basically, I know we are all equivalent genetically, forget it. But I do think it has, this is where pedagogy becomes so critical. I think that it has to do with preferential access to thoughtful people who can identify representations that work for you, right? I mean, if everyone has to be spoon-fed the same representation, it's not going to work. And so I don't think it's anything to do with genetics or anything to do with evolution, because we've reached the point where genetically we are effectively isomorphic, at least with respect to the genes that contribute to the development of the nervous system. There are differences in disease susceptibility, but not, as far as we know, in terms of cognition. Oh, right at the back. Okay, got it. Yeah.
It's just an interesting observation. A game that, like when I go shopping, if I see everything that goes in the cart, I can tell what the total cost is within a few cents. But I'm not complicated.
Mm-hmm.
Sometimes you'll think it's going on in the background.
Mm-hmm.
It's just something that happens.
Sure.
The only thing I can figure is when I was in college and had, $20 or $30 per week of groceries, I figured out everything exactly. But I'm not going through that process now. And I can say, within a few cents, I can.
No, but you're giving an example of a regimen of very strict training, which in your case had to do with various frugal considerations, which are the analog of this kid being trained by his sensei, who'd be beaten senseless if he didn't work effectively on his abacus. So, and again, right, you did internalize it, make it almost a subconscious process, which I think is one of the hallmarks of reasoning carefully. So I would agree. But before we all go, I just want to remind you that tomorrow is so much more exciting. And so I hope to see you again. Good night.